Hello, my name's Hamish and I'm a PhD student in Professor Beverly Glover's lab in the Department of Plant Sciences here at the University of Cambridge. Today I'm going to be showing you some of my research which is using the research area here at Cambridge University Botanic Garden. This is somewhere that the scientists are usually allowed to go and play with experiments, but the general public are kept out. So let's go and have a look at what we've got in there now. Come on. So I'm working inside this polytunnel, but before we go in and have a look, I want to tell you a bit about why I'm doing this research. About a third of the food that we eat depends on insect pollination to some extent. And insect pollination is where an animal like a bee or a fly visits a flower, collects pollen and takes it to another flower to fertilise it and help it grow food for us to eat. Now, as the world population grows, we're going to need to find more food, so we're going to need more insect pollination. But around the world, insects are generally in decline, and those two things don't really square up. We're going to need more insects for pollination, but there aren't so many of them around now. So, while zoologists and ecologists are working on ways to reverse this insect decline, I'm looking at the other side of things. How can we make plants more efficient for insects to find and more rewarding when they get there? And this experiment which I'm doing here is looking at the more rewarding side of things. I'm trying to find out how much nectar and pollen different varieties of strawberry make, and then that will let us know whether we can breed for varieties which make more nectar or more pollen. But first of all, we need to know this variation that exists naturally. So have a bit of a think. How many insects you might be expecting to find inside this polytunnel where I'm doing my experiment? Have a think about that, and then we'll go inside and have a look. Trip over that. There we go. So that was actually a bit of a trick question. Now we're inside the polytunnel. You can possibly see or hear there aren't any insects in here at all. And that's because I've got insect-proof netting all the way around the sides and all over the doors as well. I don't want any insects in here at all, because what I want to find out is how much nectar and pollen these strawberry plants can make when there's, and the only way to do that is to measure it, and if there's insects inside who are eating it, then I'm not going to find out how much it is, because the insects will have come and taken it away already. This is a problem I discovered in the field last year. So I've put up this polytunnel here in the Botanic Garden to be completely insect-proof, and I can grow my plants in here and then measure the nectar and pollen that they produce. To do this, on the first day that I'm doing the experiment, I will mark the I will pick all of the flowers which are currently open, because I don't know how long they've been open. On the second day, I will then mark all the flowers which are now open, so I know that they've been open for a maximum of 24 hours. And then on the third day, I'll pick those plants which I've labelled, and then I'll take them back here to my microscope, and I will measure what's going on in there. To do that, I pick all of the stamens from the plant. The stamens are the bit of the flower where the pollen is made. Put them in a tube and I stain them with a purple stain. And then back in the lab I can count how many pollen grains there are. And because, that will tell me how many pollen grains there are per flower. But also what I do while I'm here, once I've picked the stamens, is use a tiny, tiny glass tube which holds 0.5 millimetre cubed. That's a really, really tiny amount. And I use that tiny glass tube to suck out the nectar from the flower. I have to do that under a microscope because they don't produce very much nectar, and it's in a slightly inaccessible place. And then when I've done that, I can measure how much nectar the plant produced, and also I can measure how sugary it is. And using those lets me calculate how much energy the plant is making. Because obviously if one plant makes one quantity of nectar which has 20% sugar in it, if another plant makes the same quantity of nectar but it's got 40% sugar in, that plant is twice as good for the bees in terms of the energy they get to bring back to the nest. So once I've measured all of those, I can see if different varieties of strawberry produce different amounts of nectar or pollen. I've got 20 different varieties of strawberry growing in here at the moment. Sadly, I'm still in the middle of the experiment, so I can't tell you what the result of that is, but hopefully at some point in the near future I'll be able to. So now I know how much nectar and how much pollen these different plants can produce. We need to see if it's useful to the insects. And to do that, we need to go back to the Department of Plant Sciences. So I'll see you back there. So I'm now in the Department of Plant Sciences here at the University of Cambridge, and I'm going to take you into the lab behind me to introduce you to some of my friends who helped me with these experiments. Let's go and have a look. So here we are inside. It's quite a small room here, and you'll see next to me I've got this little green box. Who do you think might be in there? Let's have a look. So this little tube here is the entrance, or exit, for a colony of bumblebees, which we keep in this cardboard box. We buy them from a company in Ashton Keynes. They arrive in a small buzzing cardboard box in the post, and we use these bumblebees to help us with these experiments and tell us what it is that they think. So I'm going to talk to you today about the nectar experiments which I've done, and also which somebody else, a previous PhD student in this lab called Emily Bales, has done. Now, bumblebees can learn to associate a visual cue, something they can see, with the quality of the reward. And to demonstrate this, I'm going to first of all get you to have a go at something. So here you can see I've got a setup where I've got 6 different coloured discs, and 3 of them are yellow and 3 of them are white. And I want you to choose one of the colours. Just choose one of the discs. And the aim is to try and get a sweet and tasty reward. You chosen them? Chosen one disc? Excellent. Okay, I'm going to now show you the reward. Well done to those of you who chose the yellow ones. Okay, let's try it again. I'll cover them all up again. Pick a colour, any colour you want. Okay, ready? I'm going to show you the rewards. Again, well done to you who chose the yellow ones. Okay, I'm going to cover them up again. Pick a disc to try to get the sugary reward. And I'll uncover them. Well done again to you who chose the yellow ones. So you'll see we're getting a pattern here that you're learning to associate the yellow discs with the sweet and sugary reward, and learning to ignore the white discs which have the reward you don't want. And it's the same with bumblebees. I can use this little card which I've got here, which has one white circle and one yellow circle printed on it, and I can put some sugary water on both of them, and on one of them I'll put a sugary water which has more sugar in it, and on the other one I'll put sugary water which has less in it. And because bumblebees can learn to visually associate the colour of the disc with the reward that they get, if they can tell the difference, and if they prefer the more sugary reward, then they'll go to the yellow disc where I've put the more sugary reward. If they can't tell the difference, they'll go half and half to each of them. And actually this is what we find. We find that bumblebees do choose a more sugary reward over a less sugary reward, all the way up to about 55% sugar within that sugary water. Above about 55% sugar, they don't seem to have a preference. There's probably a really interesting reason for this, and I'm going to save that for another time, so you have to come back another year and find out more about that. So, what's all this got to do with the strawberries, which I was showing you earlier? Well, when we were doing these, when Emily was doing this experiment to have a look at what sugar concentration the bees preferred, she was using 15 cubic millimetres, which is a drop about, let's say, about a hemisphere about 3 millimetres wide on these discs. That is an awful lot of nectar. And if you remember, I said my strawberries only produce about half to 1 cubic millimetre of nectar. So I wanted to know, do bees actually still care at these teeny tiny volumes which I'm measuring in strawberries? Because if they don't, there is no point at all in trying to improve the nectar concentration within the strawberries to try to get my bees, or try to get insects to go and visit those strawberries and learn that they have a better one. If they don't care, it's not worth doing. So I wanted to see whether I could repeat this experiment with just 1 cubic millimetre of sugary water. And it turns out that doing that is really difficult, because a bee can drink that in about 2.5 seconds. Bees are also quite fickle creatures. They get bored quite easily. Now, it takes me about half a minute, or when I first started out, it took me about half a minute to reset this experiment, to put another 1 cubic millimetre of sugar water onto the card. And the bee just got bored and flew off. So I needed to work out a way of doing it faster.
It took about 6 months on and off to work out how to do it, and in the end I settled upon this absolutely hellish contraption, which is it may best be described as a bodge job. But the thing is, it works. What it is two pipettes, so scientific dispensers, which will dispense really accurate volumes, and I've gaffer taped them to a bit of floorboards. And they are spaced just the right amount apart that I can put a card underneath them. You can see it at the bottom. And then there's 2 triggers on them, and if I press those triggers, they squirt out exactly the right volume. And with a bit of practice, I could get this down to about every 5 seconds. And that is fast enough to keep the bee interested. So what I did was I put a card under, squirted the sugar on, and put the card into a flight arena. So I've got one here. This is a sort of green. It's a box with a green floor, and we put a lid on it, and the bee's coming through that hole at the end there. And I could put one card into the flight arena every 5 or 10 seconds or so, and the bee was interested enough in that. And then I could also record which colour she went to. Was it the yellow one for the more sugary solution, or was it the white one for the less sugary solution? It was a bit of a scrabble to do it, I will freely admit. The floor ended up covered in cards as I was discarding them, but I was able to do the experiment, and I was able to show that yes, bees do still care about a sugar difference even at 1 millimetre cube volumes. And that's really cool, because it shows that the experiments that we've been doing here do actually have a real-world application. And that was something which is really, really important to me when I'm doing my PhD. I want to try to do something which will have a real-world use and possibly even change the world, make it a slightly better place for my daughter. So I hope that's explained a bit about what it is that I do, and I hope you've enjoyed seeing a bit about behind the scenes in both my polytunnel and the bee lab here in Cambridge. If you'd like to find out more, then have a look at either the Department of Plant Science's website, or if you go online and search for a video called Improving Flowers to Help Feed the World. That's a video which myself and a friend put together a couple of years ago, which explains our research in a bit more detail. Thanks ever so much for listening. Do take care, and if you have any questions, then just drop me a note. I'm Hamish Symington on Twitter, do drop me a note there, or like I say, go and have a look at the Plant Sciences website. Take care, thanks for coming.